So welcome to this 10th lecture on R series, and this will be the last of the initial lectures on R series, the introduction to R series. And today we talk of how to use the dplyr library for analysis of data. So dplyr is a powerful R package to manipulate, clean, and summarize unstructured data. And this process is known as data wrangling. So to use dplyr, you need to first install dplyr into your R Studio or your R command line. So for that, you could go to the console and write install.packages dplyr. This will lead to the installation of dplyr into your R. And then to invoke dplyr into your program, you have to write library and then the library name, which is dplyr here, right? Installation you have to do only once. And once the installation is done, every time you need to call the program, you can directly write library dplyr, and that will be called into your current script. All right, so now what is the advantage of using dplyr? So dplyr gives you several types of verbs that can be used to manipulate the data. For example, you could use select to select columns. You could use filter to filter the data by certain value in the rows. You could also arrange the data in terms of ascending order, descending order, alphabetical order using the arrange command. You could create a new column with a new data. This can be done using the command called mutate. And then you could also group your data into certain groups based on certain criteria. And then you could generate your summary of your data using the summarise command. So these are some of the common verbs that are used in dplyr. So another important part of dplyr is the pipe operator, which is represented as %, greater than %. The pipe operator is used to basically link multiple operations into a single command line, right? So what is the utility of pipe operator? So whenever there are multiple operations to be performed on a base data, to get a final result, the operations will be put together into a sequence using the pipe operator, right? So basically you can do first filter, then select, then arrange, then group by, and so on and so forth. And it will basically do it in a sequential manner and give you the final result. So every step until the final step will be linked by a pipe. And I'll show you the example as we move along and you'll understand better. So we now move on to R Studio and start working with the script. So here we are in the R Studio, and we can start first with setting up the working directory. So working directory, you can set up by giving the direct command that is called setwd and the path where your file is. So I say setwd and then round brackets, and then in double inverted commas, the drive in which my folder is. So this is an F drive, and then colon followed by the folder in which my file is. So this is your R human genome folder, right? You could also do this through the session. So you could go to session, and in session, you could go to set working directory, and then you can also choose the directory here. For example, here again, if I go to F and I choose R_human genome, right? So this is the folder I'm choosing, and I say open, and you see it is coming in the console, and the working directory is set to R human genome. So this is basically to show you the advantage of using the integrated development environment like R Studio. Now we move on, and we first, of course, we need to install the libraries that are required. So as I've already told you, in this lecture, we're going to use dplyr. So the dplyr library needs to be installed into your system. If it is not installed into your system, you need to go and install it first. So I say install.packages, and then within round brackets and inverted commas, I give the library name that I want to use. So this is going to be dplyr. And then I'm going to read an Excel file. So I also require another library through which I can read the Excel file. So that is also to be installed. So what I'm installing here is the read Excel library, right? So again, install.packages, and then you say read Excel, right? So this is going to install the two packages that are required for you to run this program. Once you've installed the first time, you don't need to install it again. So in my case, I've already installed, so I'm not installing it again, but I need to invoke them into my program or call them into my script. So for that, I mentioned library, and then within the bracket, the name of the library that I want to call here. So I say library read Excel, and likewise, I say library dplyr. So these are the two libraries that we're going to use to analyze the data that is coming to us. So in the first part, what we've done is first we've defined the working directory. This is going to be the directory where my data file is. Otherwise, if I don't define my working directory, every time I want to read the file, I have to give the file path. So it is advantageous always to define a working directory. Then I have installed the packages that I require. In this case, I already have dplyr and read Excel installed, so I'm just commenting them. And then I'm invoking the libraries into my script. So I'm saying I require library read Excel and I require library dplyr, right? Then we move on and load the data first. So we load the data here. So I say HG or human genome because this data is regarding the human genome. So I say HG or human genome equals to read underscore Excel, which is a function of the read Excel library. And then in the brackets, I directly give the name of the file that I want to read. So here in this case, this is human genome.xlsx. Remember, the extension has to be mentioned, right? And then I say view HG, which is going to show me the data that was there in the file has been read into the HG variable or not. So when you run this part now, you run here and you see here it is going to, because you use the view command, it is directly going to load the file here and show you in the view window. So this is the data that we have. If you see, the first column is the chromosome, and we have chromosomes of 1, 2, 3, 4, 5, 6 until 22, which are your autosomes. Then you have the X chromosome and Y chromosome, which are your sex chromosomes, and we also have data on the mitochondrial DNA. What is the data that we have here? We have the genome size in terms of base pairs. What is the size of each chromosome? We have the number of protein coding genes. We have the number of pseudo genes in each chromosome. We have the total long non-coding RNAs in each chromosome, total small non-coding RNA in each chromosome, total microRNAs reported in each chromosome, and total ribosomal RNA reported in each chromosome. So this is basically a numerical data that we have regarding the size, the protein coding genes, the pseudo genes, the total number of long non-coding RNAs, total number of small non-coding RNAs, microRNAs, and rRNA in each of the chromosomes that I mentioned. And in terms of chromosomes, we have three sets. We have all the autosomes, 1 to 22. We have X and Y chromosome, which are your sex chromosomes, and you have the mitochondrial DNA as well, right? So this is the data that we have, and now based on this data, we're going to ask certain questions and try and find answers to them using dplyr. So let's now move on to the actual part, and we ask question number 1. The question number 1 is, which chromosome has maximum size? And the dplyr verbs that we're going to use include select, then we'll arrange it in the descending order, and then we'll pull out the first value using slice. So these are the verbs that we're going to use from dplyr. So here you are. First, what we do is we equate our base data into a temporary data that is HG1. So we say HG1 equals to HG. And then we indicate that there are further operations that are going to be done on this data by using the pipe operator. So this is percent greater than percent. If you are having trouble typing it, you can use Control Shift and M for Mumbai or M for Moscow. So we have transferred the human genome data from HG into HG1, right? So because we're looking for a chromosome with maximum base pairs, so we'll select only 2 columns here. So we use select, and we select columns chromosome and base pairs. And then we also use pipe again because we're going to further arrange this into the descending order. So we move on and we talk and we say arrange, and the order in which we're arranging is the descending because we want to pull out the first value that will have the maximum value in terms of base pairs. And the column we're using for arranging in descending order is the base pairs, right? So therefore, this is the command to arrange the data in descending order based on the base pairs. And because we're going to further perform operations on this, we use again the pipe operator. We move on and we pull out the first value by using slice, and the number 1 indicates that the first value in slice is to be pulled out. So the chromosome with maximum size is at the top. So we are pulling out the first value here. And then we say view HG1, which is going to give us the chromosome with maximum size. So when you run this here is your answer. So the biggest chromosome with the maximum number of nucleotides is basically chromosome 1. And you know, in human genome, the numbering of the chromosomes has been done in a way where the first chromosome is the biggest, 22 chromosome, which is amongst the autosomes is the smallest. So now we come to question number 2, and you can see question number 2 is not completely visible. So what we'll do is we'll do a soft wrap so that the question is completely visible within the current frame itself. So you go to code and you say soft wrap round lines. So now if you see, the question number 2 is list of chromosomes with more than 500 pseudogenes and more than 1000 protein coding genes. So for this, of course, if you were to do it in Excel, you would use filter. So same here in dplyr, there is a function called filter that can be used. So again, we equate the human genome data to a new variable that is HG2 here. And then we use pipe to indicate that there are further operations to be done over HG2. What is the further operation that we're doing on HG2? We are using a filter verb here, which is basically a verb from dplyr that allows you to filter out certain values based on the criteria that you mentioned. So here in this case, our criteria is that the pseudogenes must be greater than 500. So we give first criteria, pseudogenes greater than 500. And then with comma, we say protein coding genes greater than 1000. So this basically is going to sort out and filter out the values where the chromosomes have pseudogenes more than 500 and protein coding genes more than 1000. And then, of course, we will comment HG2 because it will show it on the console also. We will directly see it on the terminal, right? So then we say view HG2, right? So this will, again, if you run it now, this will give you a second view here, and that would basically include all chromosomes that contain pseudogenes more than 500 and protein coding genes more than 1000. So when you run this here, again, you come to view, and these are your chromosomes that contain pseudogenes more than 500. If you see here, all these chromosomes have pseudogenes more than 500. The least count that you have is 512, and protein coding genes are more than 1000. And again, if you see, the least count for protein coding genes is, I think, 1034, right? And again, as you can also see that these are not in order. So 1, 2, 3, and then 6, 11, 12, 17, and 19. So you have 8 such chromosomes in your data that contain more than 500 pseudogenes and more than 1000 protein coding genes in the human genome, right? So we now move on to question number 3, and question number 3 is to basically which 10 chromosomes have the highest protein coding gene density or genes per megabase, right? So we are looking at now a new metric that we have to calculate, that is genes per megabase. And to calculate this metric, we can use the dplyr verb called mutate, right? So mutate is able to help you create new columns. And in this column, we name it genes per megabase. We are calculating the values of the number of protein coding genes divided by the total number of base pairs in the chromosome, and further divided by 1 million because we are looking at genes per megabases. And then we say view HG3. So now when you run this here, you are basically looking at the HG3 now. HG3 contains a new additional column that is genes per megabase. So if you see here, now you have a new column that is genes per megabase, and this is the calculation of genes per megabase for each of the chromosomes present, right? So this is for chromosome 1, 2, 3, 4, 5, 6, and finally to X, Y, and the mitochondrial chromosome. But our job is not yet done because we want to look at the first 10 chromosomes with highest gene density. So we now equate the HG3, which is basically containing an additional column called genes per megabases, to HG4. And we also indicate a pipe, which means that we're going to further perform operations on this. Next, what we use is arrange. And again, we are arranging in the descending order. And the column we're using to arrange our data is genes per megabases. And because we're going to further perform operations on this, we use a pipe again, right? Now, because we only want chromosomes and the genes per megabase, so from this data now, we select specifically the columns chromosome and genes per megabase. And then we further use a pipe because we are now going to slice out this data and select only the first 10 chromosomes, which are now arranged in the descending order. And then we say view HG4. So this will now give us the first 10 chromosomes with highest genes per megabase or highest gene density. Let me just run it here, and you run. And this is the data you get. For autosomes, it is chromosome 19 that has the highest gene density, right? So when you normalize with respect to size of the chromosome, what you find is that the maximum number of chromosomes per megabase of the genome are present in chromosome 19, 17, 16, 11, 22, 20. And then comes chromosome number 1, which is the largest in size, right? So this is, again, it gives you more information on the number of genes per megabases of the genome, right? Now we ask a final question, and the final question is, calculate the mean of gene density for autosomes, sex chromosomes, and mitochondrial genomes. So what you want to do now is to calculate the mean of gene density specifically for autosomes, then for sex chromosomes, and for mitochondrial genomes. So now we know we have 22 autosomes. We have 2 sex chromosomes, that is X and Y, and we have only 1 mitochondrial genome. So, and in the original data, if you see, there is no indication that what is a sex chromosome, what is an autosome, and so on and so forth. So let me first give you a glimpse of the original data. So we say view HG, right? So if you remember in the original data, there is no indication of what is a sex chromosome and what is an autosome, except that we know that mtDNA means mitochondrial DNA and X and Y are sex chromosomes. So we need to create an additional column here so as to be able to classify chromosomes from 1 to 22 as autosomes, and then the X and Y as sex chromosome and mitochondrial DNA as mitochondrial DNA, right? So what we'll do is, again, we'll use mutate first and create an additional column that contains the notation for each chromosome, whether it is an autosome or a sex chromosome or a mitochondrial DNA. So what we're doing is, first we equate HG3 to HG5. Remember why we're using HG3? Because HG3 contains the genes per megabase data, which is not present in the original human genome data that we had read. So therefore, HG3 is now equated to HG5 because we need genes per megabase also as our additional feature. We create an additional column using mutate, where we add the notation of whether the chromosome is an autosome or a sex chromosome or a mitochondrial chromosome. So this is the vector that we're adding here. And then we say view HG5. So here you are. So this is now your additional column. This is called type. And in type, if you see 1 to 22 is your autosomes. This is your 22 here. This is your autosomes A. And then the X and Y are your sex chromosomes. And then you have the mitochondrial DNA here. Right? This is basically to use dplyr to group our data into autosomes, sex chromosomes, and mitochondrial genome. Right? What we do now is we equate HG5 to HG6 and we say pipe because there are additional operations to be done. And what we now do is we use a verb called group_by. And what are we grouping our data by? We are grouping our data by the value that is present in type. So chromosomes 1 to 22 are all A type, so they will be grouped into one set of data. Chromosome X and Y will be the second type of data, that will be your sex chromosomes and are basically marked as S, and mitochondrial genome is marked as M, so that will be your third set of data here, right? So here you are, we group by type, and then again we use the pipe to indicate that there is further operation to be done here. And what we do now is we do the final verb, that is summarize, and in summarize we say the summarize, we are basically summarizing the gene density and we're calculating the mean of the genes per megabase column, right? Based on the grouping that is done based on A, S, and M, right? So this is your summarize function, and then finally we say view HG6 to get the final data here. So let me run this part again, and we should be able to see this. So here you are, you run it, and this is what you have. For autosomes, the gene density and the average gene density is 7.52. For mitochondrial chromosome, which is just one, it is 785.5, 784.5, and for sex chromosomes, it is 3.31, right? So mitochondria, of course, is an exception in the sense that there are very small number of genes and there is no intergenic space, hardly any intergenic space. So therefore, the gene density is extremely high. But in case of autosomes and sex chromosomes, you can see that the gene density, average gene density of autosomes is more than what is present on the sex chromosomes. So these are some of the derivations you can derive through this data. And what I've tried to do is to basically give you an idea of how dplyr works and how basically you can use it to manipulate or pull out the information that you want to pull out from your data. So you could also create new columns, you could also create new calculations, and so on and so forth, as has been shown in the course of this video. All right, so we stop here, and I hope you have understood how to use dplyr. And also, I would like to request you to please like and share the videos if you truly like them. And because ultimately, it is also important that you keep me motivated and keep me driven enough to make more videos. In the previous video, one of you had mentioned that you'd also want some videos on Python. So very soon I'll be starting a basic series in Python and an advanced series in R. Thank you very much.